chemical reactions in cells are very tightly controlled. And this is a really efficient way to run an organism and run a cell. And you know some examples of this already. So for example, you know that if you eat too many calories and you don't exercise, then you're going to have too much food calories for what you're using. So you have more product than you need for that pathway, and so your body will slow down that pathway and send those calories back to making fat, right? On the other hand, if you're eating those calories and you're exercising a lot, you're working out, your body is going to start directing the energy in those calories towards building muscle. So your body is fine-tuning and controlling the chemical reactions based on what is happening in the body and in the cells. So we're going to talk about the ways that some ways that the cell does this, or that the organism does this. So the first example of how the cell controls these pathways is gene regulation. So enzymes are proteins, and so remember that means that the instructions for building those enzymes is in the DNA. So the DNA is going to be read to make enzymes. And within the DNA, there's a specific gene, so maybe gene X, that will be read to make enzyme X. And so if the cell turns off gene X, that means it can't read gene X, then, so let me write this here, turn off the gene, then that will decrease the amount of enzyme X. So what will that do to the chemical reaction? It will slow it down. If, on the other hand, I then turn the gene back on, it will increase the amount of enzyme, and then that will speed it up. So that will increase the rate or speed it up. So by turning on or off genes, I can increase or decrease the amount of enzyme, and by doing so, I then either speed up or slow down the chemical reaction. So I think I said that the reverse way that time. So decrease enzyme slows it down, increase enzyme speeds it up. Another type of regulation is cellular regulation. This is when cells receive signals from their environment and adjust their chemical reactions. So the classic example of this is endocrine signaling, which we've talked about in a previous chapter. And so in that case, hormones that are secreted somewhere in the body can distribute in the bloodstream, and the cell will receive that signal and change its chemical reactions. A classic example of this is what happens when you get scared. So when, say, someone jumps out at you, you have this feeling in you, or rather you have this response in your body that's the release of a hormone. And that hormone travels through your body and it changes the chemical reactions that are happening. It speeds up the breaking down of calories because it's preparing you to either fight or fly. So it's called the fight or flight response, right? And so what you do is you increase your metabolism to prepare to either run or fight. And so the feeling in your body is an increased heart rate, increased breathing, and that is all a result of the increased metabolism. So the cell receives the signal, that's the adrenaline that was released in your body, and then it speeds up the chemical reactions, and you can feel that happening because your heart rate goes up and your breathing increases. The third type of regulation is biochemical regulation. This is when molecules will actually attach to the enzyme and change how well the enzyme works. We're going to focus on inhibitors. So to inhibit means to slow down the enzyme. So inhibitors slow down enzyme activity. We'll look at two examples that work in very different ways. Competitive inhibitors get that name because they compete with substrate for the active site, and non-competitive inhibitors don't bind to the active site, so they don't compete for the active site. Instead, they're going to bind to something called the allosteric site that causes a shape change. Another example, or a more specific example of non-competitive inhibitor, is feedback inhibition, and this is where a product of the pathway inhibits an earlier step in the pathway. So we're going to walk through these three now in a little bit of greater detail and look at some figures. So again, a competitive inhibitor binds to the active site of an enzyme. And so it competes for that active site. If it gets in there first, then the substrate can't get in. So this is the normal enzyme, and see, this is its special shape, that it's unique for the substrate. So the substrate perfectly fits into there, and then if it gets in there, a chemical reaction will occur. The competitive inhibitor, if it gets it in there first, can block the substrate from binding. So it's actually sitting in the active site. As long as it's there, the substrate can't get in, so no chemical reaction will occur. Now, these are usually reversible. And so that means that eventually this guy will float away. And once he floats away, then the substrate will be able to get into that active site and a reaction can occur. But again, and the competitive inhibitor will slow it down as long as it's in that active site. So again, before I move on to the next slide, I just want to remind you, this is what my active site looks like. That's what my substrate looks like. Because if I look at a non-competitive inhibitor, in that case, the inhibitor is down here. Oops. The inhibitor is down here, and it's binding to the allosteric site, somewhere else on the enzyme. And when it binds there, it changes the shape of the active site. So now this active site doesn't fit the substrate. So now the substrate doesn't fit. If the substrate doesn't fit, then there will be no enzyme activity, no chemical reaction. So this is a great example of how structure equals function. So by changing the structure, this enzyme is no longer able to carry out its reaction because it's not binding its substrate. Again, this is usually reversible. So eventually the inhibitor will leave the allosteric site and the enzyme will go back to its original shape. So the enzyme can go back to its original shape and carry out the reaction. Feedback inhibition is a special type of non-competitive inhibition where a product of the whole pathway will inhibit a different step in the pathway. So if I look at this example, there's 3 steps to the chemical reaction. This is my final product, and my final product can actually go back to enzyme 2 and bind to enzyme 2 in an allosteric site. And when it binds to it, now the substrate won't attach. So this is a great way to manage your chemical reactions if you think about this as a factory. Imagine you're running a car factory, right? So you're starting with raw materials, and through a series of steps, you're going to turn that into a finished car. And then somewhere down the line, many, many steps down, somebody is buying that car and taking it off your hands, right? If nobody is buying your cars, right, you'll start to get lots and lots of cars built up. Too many. And do you want to keep making cars if you're not selling cars, right? So really maybe say selling here, right? So selling cars is normally getting rid of the cars that you're making. If you're not selling them, that means you're getting stuck with lots and lots of accumulating cars. So in the factory, you want to stop making the cars. So you want to go back and say, no, don't make the cars anymore, right? So then your inventory would start going down to match the pace at which you were selling the cars. So in a factory, a manager would have to be making this decision based on car sales and things like that. The cell has it automated so that the final product simply is able to do the amount of final product. So the higher this final product gets, the more it will come back and do this. And so the more final product I have, the more it will slow down the pathway so that I don't make too much of my product. So, I'm sure that factory managers would love to do it this way too, but because the cell is so efficient at determining how much product it should make. So So again, feedback inhibition is when the product directly goes back and tells the pathway to slow down because there's too much product. So we'll sort of wrap up by looking at a question here about feedback inhibition. So this is showing an actual chemical pathway. The end product is an amino acid called isoleucine. And in this case, isoleucine feeds back to the original enzyme and it binds to an allosteric site and changes the active site. So if isoleucine levels are high, then this will bind more and this will decrease the rate of this pathway. If isoleucine levels are low, right, so now I have not enough product, then I won't get this happening. So I won't have any inhibition. And so this will speed up the rate of the pathway, right? So again, this is fine-tuning and automating this whole process. If I have too much product, I will slow down the production of the product. If I don't have enough product, then I will speed up the rate of the product. This is a nice way to regulate chemical reactions in the cell.